Welcome back. I hope that you're keeping up with us and finding this material interesting. I know that we're delving into a lot of basic science, but I really believe that there are a lot of people hungry for more than a superficial look. And so we will now delve a little bit deeper into protein chemistry, and this will allow us to see that the chance evolution of functional proteins is actually even much less likely than we have already established. Okay, so let us just take a step back. We said that the information in DNA is about the correct sequence of amino acids to make a protein. Our previous analysis didn't even talk about finding the correct sequence of amino acids to make a particular protein. It simply said that if I have a modest length protein of 150 amino acids, the likelihood that they would all be L amino acids joined by peptide bonds is unimaginably small if this were taking place by chance, the chance theory of prebiotic evolution. But now we come to the real magic, if you will, which is that not only do I need to have L amino acids joined by peptide bonds, but I need the correct sequence of amino acids. And that is where the real information of DNA is in fact stored. That's where we can fathom the enormity of the information. Why? Because so far we've simply talked about proteins that if I get the correct sequence of amino acids, I'm done. But why is that correct sequence so important? It is because proteins don't stay as a chain of amino acids. This chain of amino acids, to be able to function in the body, has to undergo a very complex folding scheme to form a three-dimensional structure. And it is that three-dimensional structure which functions in the body. Just simply a string like pearls on a pearl necklace in and of itself does very little good. So once that correct string of amino acids is specified, it will undergo one of two types of folds. It will twist itself into an alpha helix or sort of into a corrugated sheet known as a pleated sheet. So once I have sections of it that are alpha helix and sections that are pleated sheets, then that chain would then undergo yet another series of folds to twist up on itself, and that is what is known as tertiary structure. The alpha helix or pleated sheets are the secondary structure. Then imagine that the chain has turned into an alpha helix, or a section of it is an alpha helix and a section is a pleated sheet. Then that folds further upon itself into a tertiary structure. And what this slide is showing is, for example, how this happens with the critical protein hemoglobin. Once I have that one chain, it may then go and function as a protein, an enzyme, for example, or it may need to combine with other chains to form the full protein. So in the example of hemoglobin, I have two chains known as alpha chains and two chains known as beta chains, and I need to fold each one of those correctly in the tertiary structure and then combine all four of them into a so-called quaternary structure. And then and only then would I have a functional protein. And so here is a schematic of the hemoglobin protein with the alpha and beta chains folded in on themselves in this very complex tertiary structure and then combined in a very complex quaternary structure to form a very complex three-dimensional shape, which then functions to transport oxygen. And we've already seen how one mistake in one amino acid throws off the entire scheme and takes functional hemoglobin and turns it into sickle cell hemoglobin, for example. So this is a very delicate process. And the information in DNA to specify the correct amino acid sequence is critical for this cascade of a correct secondary structure, tertiary structure, and quaternary structure. Now, to understand just how significant this is, I've pulled an excerpt from Professor Albert's book, Molecular Biology of the Cell, because for a protein to function in our bodies, when we make it, it must fold correctly and assume this very complex three-dimensional structure, which must be stable. And it must do that over and over and over each time we make it. So imagine making the hemoglobin molecule and stringing together a long chain of amino acids. Now, if you've ever had a telephone with a long cord, or been out doing some fishing with fishing line, or trying to untangle your hose in the backyard to water a tree, you realize that these long chains can end up getting folded in a whole bunch of different ways. And it's very hard to get the same folding twice. When things get knotted up, they don't normally knot up in the same way. But our long strings of amino acids need to fold in the exact same way every time and in the correct way, and then have that folded configuration be very stable. And all of this results from the correct sequence of amino acids. If I just start putting amino acids together at random, it becomes nearly impossible to find a string of amino acids that will always fold in the same way over and over again, and that resulting three-dimensional structure would remain stable. So let us read together this excerpt from Professor Albert's book. So he's saying that since each of the 20 amino acids is chemically distinct and each can in principle occur at any position in a protein chain, there are 20 times 20 times 20 times 20, which is 160,000 different possible polypeptide chains 4 amino acids long. Now, if we want n amino acids, like 150, for example, so let's keep reading together, or 20 to the n different possible polypeptide chains, n amino acids long. For a typical protein length of about 300 amino acids, more than 10 to the 390. That's an unimaginably large number, and the way we get that is that is 20 to the 300, 20 times 20 times 20, 300 times to fill in the correct amino acid sequence. So there are 10 to the 390 different polypeptide chains that could theoretically be made. This is such an enormous number that to produce just one molecule of each kind would require many more atoms than exist in the universe. Now, of this extremely large set of possibilities, let's keep reading. Only a very small fraction of this vast set of conceivable polypeptide chains would adopt a single, stable, three-dimensional conformation. By some estimates, less than 1 in a billion. The vast majority of possible protein molecules could adopt many conformations of roughly equal stability, each conformation having different chemical properties. And yet virtually all proteins present in cells adopt unique and stable conformations. How is this possible? And that really is the information enigma. the DNA specifies a sequence of amino acids that out of the vast set of possible sequences adopts a single stable three-dimensional conformation that is able to perform a biological function rather than flip from one conformation to another, rather than get knotted up into some useless three-dimensional shape. And these protein knots, by the way, are a very real hazard. When proteins don't form correctly and knot up, this is, for example, the basis of degenerative diseases in the brain like Alzheimer's disease, where tau proteins form so-called neurofibrillary tangles, knots of protein that destabilize the functioning of a cell. It is also these abnormal protein folds that are at the heart of so-called prion diseases of the brain, very scary, rapidly progressive dementias like Jakob-Creutzfeldt disease. So having stable, functional, unique folding patterns is critical, and you see how extraordinarily unlikely this is to occur if we were to simply string together amino acids by chance. And Professor Alberts gives an estimate of less than 1 in a billion. We will see that the likelihood is actually much, much, much less than that. So the relevant work here was done by Dr. Douglas Axe, a protein chemist, and he was working at Cambridge University at the time. And his important results were published in the Journal of Molecular Biology in 2004. And what he set out to do was to estimate what is the probability of finding a functional protein in the so-called amino acid sequence space. If you were to randomly string together amino acids, what is the likelihood that they would form a functional protein? Now, he was looking at one specific protein, a beta-lactamase enzyme, and he found that the odds of getting this protein were not 1 in a billion, they were actually 1 in 10 to the 77. That's 1 in 1 followed by 77 zeros. So 1 in more than a million, trillion, 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 etc. So exceedingly small. But you may ask, well, this is not really fair, because of course it will be very hard to form one specific protein that we're looking at. How about what is the likelihood of forming any sort of functional protein, any useful protein? Because that's really the critical question to the origin of life. And his work allowed a rough estimate of that as well, and judged by, as we said, a protein that would form a single, stable, unique, three-dimensional configuration through its folding pattern. And his estimate was that it would be roughly 1 in 10 to the 74. So a bit more likely, 1,000 times more likely than getting the specific protein he was after, but still it is exceedingly rare for random sequences of amino acids to be able to form functional proteins, much more rare than what Professor Alberts sort of said in an off-the-cuff fashion. So we see that this is a truly rare situation. So Axe's work now makes it possible to calculate the odds of a functional protein and to see how really rare that is. And that is exactly what Stephen Meyer does in Chapter 9 of his book, Signature in the Cell. So let us go ahead and read the relevant section together. Quote, the odds are prohibitively stacked against a random process producing functional proteins. Functional proteins are exceedingly rare among all the possible combinations of amino acids. X's improved estimate of how rare functional proteins are within sequence space has now made it possible to calculate the probability that a 150 amino acid compound assembled by random interactions in a prebiotic soup would be a functional protein. This calculation can be made by multiplying the three independent probabilities by one another. The probability of incorporating only peptide bonds, which we've already seen is about 1 in 10 to the 45. The probability of incorporating only left-handed or L amino acids, 1 in 10 to the 45. And the probability of achieving correct amino acid sequencing, and this is where X's work of 1 in 10 to the 74 comes in. Making that calculation, multiplying the separate probabilities by adding their exponents, we would get a dramatic answer. The odds of getting even one functional protein of modest length, 150 amino acids, by chance from a prebiotic soup is no better than 1 chance in 10 to the 164. So let us end this lecture by continuing with Stephen Meyer's discourse. Quote, it is impossible to convey what this number represents, but let me try. With the odds standing at 1 chance in 10 to the 164 of finding a functional protein among the possible 150 amino acid compounds, the probability is 84 orders of magnitude or powers of 10 smaller than the probability of finding the marked particle in the whole universe. Another way to say that is the probability of finding a functional protein by chance alone is a trillion, 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 trillion times smaller than the odds of finding a single specified particle among all the particles in the universe. So this should really be the end of the discussion on the possibility that proteins and life arose by chance, and it should give us an idea of the immense amount of information that is being specified and stored by DNA, and we will see that actually the story continues.